Hi, everybody. I'd like to tell you a little bit about the research that's been done in my laboratory with funds from a very special group called Students Supporting Brain Tumor Research. First, just a little bit about SSBTR. This is the largest student-run nonprofit organization in Arizona. It was founded in 2002 after 3 local high school students were diagnosed with and ultimately died from their brain tumors. They've raised an astonishing amount of money, more than $3.4 million to date, for research. Students from over 100 schools have participated, and about 93% of every dollar raised goes directly to local and some national organizations that are involved in brain tumor research. So if you'd like more information, or if you'd like to donate or register for their signature event or buy from a truly excellent silent auction, you can use these web links that I have here, and I'll repeat them at the end of my talk. So just very briefly, if you're not very familiar with brain tumors, the worst grade of adult brain tumors, called glioblastoma or grade 4 astrocytoma, the standard of care is the maximum amount of surgical resection that can be done, followed by radiation and chemotherapy. And there are few options available other than that. Average survival is about 16 to 18 months. The tumor does typically recur within this time period, and when the tumor does recur, it often doesn't respond very well to additional therapies. Having said that, there are numerous clinical trials with new therapies that are being done now and additional trials that are always on the horizon for the future. In addition, brain tumors are the leading cause of death due to solid cancers or solid tumors in children. If you look at survival of children with brain tumors, and this is some information I got from the Max Love Project, unfortunately, even when children survive brain tumors, they still have issues. So there's increased risk of death from other causes. There's an increased risk of various long-term side effects. So clearly this is a disease that really needs a lot more work. So what is cancer? Well, in 2011, Hanahan and Weinberg came out with a revised version of a paper they wrote called The Hallmarks of Cancer. And that picture that you see to the left is actually a picture of a brain tumor. So what are the hallmarks of cancer? First of all, the tumor cells are immortal. In other words, they will continue to grow as opposed to the rest of the cells in our body, which generally die of old age after a while. The tumor can avoid destruction by the immune system. The cells can resist cell death. What I mean by that is, under normal circumstances, the cells in our body have a mechanism whereby they will die if they are damaged. So if, for example, your DNA is damaged by sunburn or chemicals or other things, the cells can sense this, and if the damage can't be repaired, the cells will die. In tumor cells, this generally doesn't happen. So even when cells are damaged, if the damage itself doesn't kill the tumor cells will continue to grow, and this helps to propagate mutations. Tumor cells induce something called angiogenesis, which is the formation of new blood vessels. So as this tumor grows, it causes the formation of new blood vessels inside the tumor, so the tumor continues to get nutrients. Tumors also can invade the tissue around them, and the cells can break off from the main tumor and travel to other parts of the body. Tumor cells have ways to continue to grow. So whereas our normal cells have signals that say grow when they need to grow and stop growing when they should stop, tumor cells simply continue to send out signals to say grow. They also tend to promote inflammation, and inflammation can enhance the growth of tumors. And the one that my laboratory is studying is called deregulating cellular energetics, which is a fancy way to say the metabolism of tumor cells is basically screwed up. But if you look at this little picture, each of these little icons represents a different hallmark of cancer. This one here is deregulated metabolism, but all of these things on the outside are various metabolic pathways or metabolic substrates that affect different hallmarks of the tumor. So instead of the deregulated metabolism only affecting the metabolism hallmark, it affects basically all the hallmarks of cancer. So metabolism is a central thing in So what about metabolism in cancer? Well, in the 1920s, Otto Warburg came up with this idea, which has been named after him, called the Warburg hypothesis. Basically what he found was that cancer cells use their glucose in a way that's different from normal cells. Glucose is a sugar molecule. It's a basic molecule of energy for our cells. And the way cancer cells use their glucose, they actually get less energy from each molecule of glucose that they use. Because of this, cancer cells need a lot of glucose and glutamine, which is another molecule that can be used for energy, more so than most of our normal cells. But in addition to needing a lot of glucose and glutamine for energy, the way they use this glucose and glutamine allows them to create the building blocks to make more cells. In other words, they're making the pieces they need to make more cells. Well, an obvious question comes from that. Since tumors use a lot of glucose, can you simply starve the tumors by reducing the glucose in the body? And like I said, metabolism affects every hallmark of cancer, so if you can alter the metabolism, perhaps you can really affect the cancer. It's a great idea. In fact, if you can affect the metabolism, you're going to affect more than just the cellular energetics of the tumor cell. However, you have to do this in a way that you're not harming the normal cells. And that's where we get to what my laboratory has been studying, which is called a ketogenic diet or metabolic ketosis. Now, most people have heard of a ketogenic diet nowadays, but they've heard of it usually in the context of weight loss or sports enhancement to enhance the performance of an athlete, or possibly to enhance cognitive activity. When we talk about the ketogenic diet as a therapy or metabolic ketosis, we're really talking about a fairly extreme version of this diet. Don't do this at home, folks. This is the kind of thing that you really want to do with the help of a dietitian if you're doing it at a therapeutic level. It's very high fat, very low carbohydrate, and low protein. It is, in fact, used to treat epilepsy when drugs don't work. In particular, it's been used for years for pediatric epilepsy. It's now being used for adult epilepsy as well. It is being looked at for the treatment of brain tumors and other diseases, particularly neurodegenerative diseases. And now I'm going to tell you a little bit about our previous work, which I'm very proud to say has been supported by SSBTR. So we've looked at the therapeutic ketogenic diet. Like I said, high fat, low carb, low protein. What does that mean? Well, here's your normal diet. It's about 55% carbs, about 15% protein, 30% fat. When we talk about sort of a normal ketogenic diet, in other words, the type of diet that a person might go on if they want to lose weight or an athlete might use, the protein is probably about 20%, fat might be 75%, carbs might be 5%, maybe a little higher. But a therapeutic ketogenic diet is very extreme. So 90% fats and about 5% carbs and about 5% protein. And this can be modified. There are modifications of the ketogenic diet that can be used for therapy. But this is the diet that we've looked at in our laboratory. What this does is it reduces the glucose in the blood, and it increases something called blood ketones. Ketones are molecules that your body makes when it metabolizes fat. And it turns out that ketones are an excellent energy source for normal cells, in particular for the normal brain. So when you reduce blood glucose, as long as those ketones are there, your normal cells are very happy. However, tumor cells cannot use ketones for energy. At least that's the general thought, that cancer cells require glucose and have more difficult time using ketones. Like I said before, this has been a known treatment for pediatric epilepsy, so it's got a safety profile that's well known in the medical community in terms of people who have studied epilepsy. It's now being looked at for other diseases such as Alzheimer's, to a lesser extent for Parkinson's, for other neurodegenerative diseases, ALS, things of that nature. But we and others have found that it does inhibit the growth of brain tumors in vivo. In vivo means in an animal model. And I am an animal lover. Any animal work we've done is extremely humane, and it has to be approved by a veterinarian and a committee of people. But there is no therapy that anybody has ever had that hasn't been tried in an animal model to prove it's safe. So having said that, there are mouse models of brain tumors, and ketogenic diet has been shown to inhibit the growth of the tumors in these mice. There are also case studies in humans that are published of 1 or 2 people that have used the ketogenic diet and done well. And there are anecdotal studies. We all know about Facebook and other social media, and you'll see a lot out there on social media. The fact that it's anecdotal doesn't necessarily mean it's right or wrong. What it means to me as a scientist is if there's enough anecdotal studies that are out there, it suggests that it's something we should look at. And in fact, there are now clinical trials that would be a more definitive proof that the ketogenic diet or metabolic ketosis actually does affect the growth of tumors. So the exciting thing about what we'll call metabolic ketosis, which is something that you get on a straight ketogenic diet, is it is extremely pluripotent, which is just a fancy word for it does a lot of things. And the way we've been studying it in my laboratory is to use it as an adjuvant therapy. In other words, we're not using it alone. We're interested in how is it going to work with the standard of care that a patient might get. And there's a number of ways that people can get into metabolic ketosis. Severe caloric restriction is one thing because a ketogenic diet actually mimics fasting in a lot of ways. Intermittent fasting is another thing that has been popular in the literature and probably has some utility for people undergoing treatment for cancer. There's something called exogenous ketones. These are available readily on Amazon. Essentially, people have taken the ketone chemical and they make it into drinks. So you can actually drink exogenous ketones and it will raise your ketone levels. When you do it that way, your ketone levels will go up short term and then come back down. There haven't been a lot of definitive studies on exogenous ketones directly with cancer, although there are some. It hasn't been looked at to a tremendous extent in combination with radiation and chemotherapy, but it is something that people are starting to think might be useful in addition to a ketogenic diet. And of course, the therapeutic ketogenic diet, as I said, is the way we've been looking at things. So here's some of the data we've gotten. First of all, when we put animals on a ketogenic diet, and we have used a human ketogenic diet that's available called KetoCal, the faster this line drops, the faster the animals are succumbing to the disease. And this is a combination of 2 experiments. So this black line is animals that are just on standard mouse chow, and the red line is animals that were getting the ketogenic diet. These animals had tumors implanted in their brains, and you see that there's a very significant increase in their survival. But as I said before, we're really interested in how this might work in a patient, and that means we have to look at it in combination with the standard of care, which includes chemotherapy. So the chemotherapy that patients get now is called temozolomide. This black line shows survival of animals getting temozolomide and standard diet. The green line is animals that have gotten temozolomide in combination with a ketogenic diet. And again, you see a very significant increase in their survival. But what was really exciting to us is when we did radiation therapy. Radiation therapy is a mainstay of brain tumor therapy for patients with malignant brain tumors. And when we did radiation with the standard diet, again, we got an increase in survival, as you would expect if the radiation is working. But when we did radiation plus the ketogenic diet, it was amazing. We actually got a cure in 9 of 11 animals. And in fact, when we switched these animals back to standard diet, the tumors did not come back. We published this. All of these notices on the bottom are actually the publications, and they're available in what's called an open access journal, which means anybody can get them. And we started to get contacted by patients and patients' families, which is a very unique experience, I think, as a scientist, that not many of us get contacted by patients. But all we can say at this point is, yes, it seems to work in mice. It doesn't prove that it will work in people. But we did start to look at mechanisms. And when we started to look at mechanisms, clearly a lot was going on that wasn't just glucose. I don't have time to show you all of the data, but I can tell you that the things that we found is that when we looked at tumors from animals that were on a ketogenic diet, we found a reduction in the factors that promote tumor growth. We found a reduction in the inflammation around the tumor. We found a reduction in the swelling around the tumor, which is very important because patients with brain tumors very often are given steroids to reduce the swelling. We found a reduction in angiogenesis. Remember, that's the formation of new blood vessels to feed the tumor. And we found that there was a reduction in hypoxia. Hypoxia means reduced oxygen. What happens in tumors is there are usually areas in the tumor that have reduced oxygen. They're called hypoxic areas. And what happens with hypoxic areas is that actually turns on a number of genes, including this one here called HIF1 alpha. And that gene then turns around and turns on a whole bunch of genes that promote the growth of the tumor. So essentially, by reducing hypoxia, you can reduce the expression of a gene that really promotes the growth of the tumor. The other thing is that the cells that are in the hypoxic areas tend to be more resistant to therapy. So if you can reduce the hypoxia, you may be able to sensitize some of these cells to some extent. We also found that the ketogenic diet reduced the expression of other genes that I would call pro-tumor. In other words, genes that are known to be turned on in cancer and that essentially start a cascade of a whole bunch of things that help the tumor grow. And also very exciting is, in collaboration with scientists at Arizona State University, we found that the animals on ketogenic diet had an enhanced anti-tumor immune response. So what are the mechanisms of action of all this? It just seems like it does so many things. It's really hard to wrap your arms around how one thing can have so many different effects. And what we think is going on is something called epigenetic modifications. Now, epigenetics is a study of changes that happen when gene expression is changed. In other words, what genes are turned on or turned off, that's changed, but not the actual gene itself. In other words, we're not talking about mutations, we're not talking about changes in the DNA itself, we're talking about changes in the way the DNA is being used. So epigenetic changes affect how the genes are essentially read by cells. If you think of genes as the words in a book, every cell reads the book, but they don't necessarily read the entire book. They read the chapters that they need to do their job. And epigenetics affects which words are read. And this affects how the genes make proteins that ultimately does the work in the cell. So one of the things we've looked at is something called microRNAs. So for those of you that are scientists in the crowd, microRNAs are non-coding RNAs. That means they don't make a protein. They're very, very small. They're 22 nucleotides long, which is tiny. When we talk about other forms of RNA, they're thousands and thousands of nucleotides long. It reduces the production of particular proteins, so they are specific to turn certain proteins off. But a single one actually might reduce the production of hundreds of proteins. So it's not one microRNA to one protein. They are a little bit promiscuous. And the repression is often not huge. It might be a small amount. But when you're talking about the control of cellular functions, things don't have to be huge to make a big change. So changing the expression of a protein twofold could indeed make a big change in how the cell acts. And microRNAs are now being looked at clinically because they have a lot of potential for early diagnosis of disease, for prognosis of disease. In other words, how well is a patient doing, how bad is the tumor, essentially, the grade of the tumor. And they're also being looked at as therapeutic molecules in cancer. So in collaboration with my excellent collaborator, Dr. Nel Syed at Imperial College in the UK, we implanted tumors in the brains of these mice. We sacrificed the mice humanely and sent the tumor tissue to her, and she did a global analysis of microRNAs. So there are lots and lots of different microRNAs, and she actually did an analysis of all of them. And what she found was that there were quite a few that were increased more than 2.5-fold, and a few were decreased. When microRNAs go up, they will turn off the expression or reduce the expression of particular proteins. So there were quite a few that were changed. If we looked at which ones were changed, and again, this is that hallmarks of cancer little pictogram, and each of these are the different hallmarks. Each of these things on the outside are classes of therapies that are being invented, tried, looked at to inhibit particular hallmarks of cancer. So for example, if you look at angiogenesis, the formation of new blood vessels, there are pharmaceuticals that inhibit some of the signaling that causes angiogenesis, just as one example. So all of these hallmarks have potential therapeutic value and potential therapies being worked on. So looking at it in a chart form, here's the symbol of the hallmark, here's the words of the hallmark. These are all microRNAs that are implicated as being involved with particular hallmarks of cancer. The ones that have red squares around them are microRNAs that are increased in tumors of animals fed KD versus standard diet. In other words, the microRNAs, the expression of the microRNAs from the tumors of animals fed a ketogenic diet was increased in just about every hallmark of cancer. So when we're talking about something as potentially simple, I guess you could say, as changing a diet, you have this tremendous ability to change lots of the things going on in tumors. And this is what's very exciting to us, because not only could the ketogenic diet reduce the growth of the tumor, but it can also enhance the activity of many different therapies because it affects many of the hallmarks for which there are therapeutic possibilities. So our most recent work, I was at Phoenix Children's Hospital until that research laboratory closed. We started looking at a particularly devastating disease called diffuse intrinsic pontine glioma, or DIPG. This begins in the brainstem, which is a part of the brain that controls breathing, heart rate, and various nerves and muscles to help a person do everything, including eat. It usually occurs between ages 5 and 7. It makes up about 10 to 15% of all brain tumors in children, which means there's 150 to 300 new cases in the US each year. Unfortunately, fewer than 10% of these children survive more than 2 years from their diagnosis, and in fact, it's pretty much considered virtually uniformly fatal. The treatment is steroids and radiation, but unfortunately the radiation is palliative. What that means is it will slow the growth of the tumor for a while, but it doesn't appear to be curative. And the therapies, the chemotherapies that have tried to date have not been very effective. So we're very, very interested in seeing whether the ketogenic diet helps this. I'll show you some, you can't do surgery on this tumor in general because of the location. So it can't be surgically removed because it's in a very, very important part of the brain. So this is just a little bit of data that we got before the laboratory closed. It's preliminary, it is not published, but these are cells that have been grown from DIPG that we received from Dr. Michelle Mongi at Stanford University. And this is just showing cell growth. And what it looks like is, so all the black lines are control, which means the cells are just grown, and that's how many cells we had after a certain number of days. When we added a ketone, the main ketone that your body makes, beta-hydroxybutyrate or BHB, it does seem to inhibit the growth of these cells to some extent. When we added radiation, which is the red bar, again, it seems to inhibit. And the fact that the amount of inhibition is different in the different cell lines is totally normal. When you look at different cancers, you very often see a difference in the way they respond to things. But what was exciting is the green line. In all the cases when we added the ketone plus the radiation, we got a better effect than either the ketone or the radiation alone. So we're very excited about that. We would like to continue working on this, and I do have some plans to continue working on this with my collaborator, Dr. Nell Syed. And we're really hoping that the ketogenic diet might start to be looked at in pediatric brain tumors. There are now people that are using it in adult brain tumors, and there are people who are doing fairly well using it. So we're super excited that Student Supporting Brain Tumor Research has supported this research, and we've gone from bench to actually affecting some patients now. So where are we going now? Well, first of all, we really need to know more about how it works. It's kind of like unraveling a sweater. The more you learn, the more there is to learn. It does many, many things. What is the best way to get this to patients? There are a lot of physicians that don't believe in this. Some do. So we are working with physicians. We're working with patients in patient groups. If somebody calls me, I can turn them on to people that can help them. Various organizations. We give national and international presentations. In fact, if you look up my name on a web browser like Google, you'll find a recording of talks that have been given at various meetings. You will also find recordings of talks of some of my colleagues that have been given at these same meetings, and you can find out more about what's being done in this arena. We go to professional meetings and present. Social media very often has things. We publish papers. Like I said, you can find papers that have been published not only by myself, but by some very excellent colleagues that have been working on this as well. We do acknowledge SSBTR for funding the work in my laboratory. It's been really instrumental in getting this going in my lab. And what I'm very excited about is clinical trials. There are some clinical trials out there. They're difficult to do, mostly because they're difficult to get funded. Ketogenic diet is food that you can get from a supermarket, so there's no incentive for a company to support clinical trials. Clinical trials are extremely expensive. But we are looking at getting some of these going. It's also hard to get patients to be compliant unless they work closely with a dietitian. So it's not the easiest thing to do, but there are some being done. We do know that it can be done safely, and it does appear to help some people. Dr. Chris Smith at the Barrow Neurological Institute is a fantastic neurosurgeon. He specializes in brain tumors and epilepsy, and he is now so totally sold on the use of a ketogenic diet. He wants all of his patients to be on the diet. His wife is a registered dietitian, and she has been working with people as well. So they are wonderful resources, and he goes to professional meetings with his neurosurgical colleagues and speaks about it. And recently we've discussed having a clinical trial with Dr. Lindsay Hoffman, who is a neuro-oncologist at Phoenix Children's Hospital. And I'm very happy to say that a group of us, including the two dietitians at Phoenix Children's Hospital, Lisa Venata and Christy Wheeler, they are the ketogenic diet dietitians at Phoenix Children's Hospital. And as a group, we are all looking at starting a pilot trial in pediatric brain tumors with the ketogenic diet. This is just getting off the ground now, but all of this really is due to the funding of SSBTR that really got us to this point. I'd like to thank the members of my laboratory. Obviously, I don't do this myself. This is just a small list of some of the people that have contributed a great deal to this work. When my lab was open, we always had high school and college students working there, and they were always incredibly helpful, and it's wonderful to actually have them involved in research. I've been very fortunate to have wonderful collaborators in this work. I've already discussed the microRNA work done by Dr. Nellifer Syed in her laboratory at Imperial College in the United Kingdom. We've had MRI imaging done by Dr. Chad Quarles and Dr. Gregory Turner at the Barrow Neurological Institute. The immunology work was done in the laboratory of Dr. Joseph Blattman by Dr. Danielle Lucier at Arizona State University. And the DIPG work was done with Dr. Cynthia Wetmore. I've also had a number of other collaborators for many aspects of this work that I was unable to show you during this talk because of time constraints. Of course, nothing can be done without funding. One of my main supporters is Students Supporting Brain Tumor Research. They are the reason I was able to begin this work on the ketogenic diet. I've also been fortunate enough to have funding by the Nicola K. Johnston Revocable Trust, the estate of Janice Nicholson, the Max Love Project, Matthew's Friends. Dr. Nil Syed has had funding from Brain Tumor Research Campaign and Brain Tumor Research in the UK. And early parts of this work were funded by Arizona Biomedical Research Commission, and Nutricia has provided the KetoCal for these studies. If you'd like more information, or if you'd like to donate to SSBTR, or if you'd like to register for the Curathon, or to purchase items from the silent auction, and they've got great items in their silent auction, please look at these websites. The registration can be found on Eventbrite, but you can also get that link off the SSBTR website. And if you have any questions, please feel free to send an email to admin@ssbtr.org. In conclusion, I want to thank Students Supporting Brain Tumor Research for their ongoing funding of this work. It has been instrumental in getting this done, and I'd like to thank you all for your attention.